Hello, this is Dr. Jabour. Today we're going to talk about muscle tissue and the physiology that takes place at the neuromuscular junction. These are the instructional objectives, and let's get going. All right, so in the beginning, I have several slides on general characteristics of muscle tissue. Whether it's cardiac, skeletal, or smooth, there are three types of muscle tissues, and the main function of muscle is movement. That's the fundamental characteristic of all living things. It's to be able to create movement, and the muscle is going to be at the source of that movement. When the cell contracts, the muscle cell, it shortens, and because these cells are attached to things, whatever it's attached to, it's going to move those structures, whether it's the dermal layer under your skin for facial expressions, or it's the bone that it's attached to, and now we're moving a bone at a joint, or whether the muscles are sphincter-like muscles, and they're squeezing content down a tube. All of this is about movement. In the process of creating that movement, we have the usage of adenosine triphosphate, which is a molecule that has potential energy in its covalent bonds. And when the phosphate group, so there are three phosphate groups attached to adenosine. When these three phosphate groups, when one of them is cleaved, there is a release of energy and also release of that phosphate, and you end up with adenosine diphosphate and the release of energy. So that provides, the release of that energy provides the mechanical energy necessary for moving the contractile proteins inside the cell, which then shrinks the cell, which then moves the structures that the cell is attached to. So these are fundamental characteristics of the three types of muscle tissues: cardiac, skeletal, and smooth. So there one slide on each muscle type, and this lecture on the physiology of contraction is mainly about the skeletal muscle tissue. So I suggest creating a little table comparing the three tissue types, unless you already have created something like that. When we reviewed muscle tissues as part of the histology chapter in a previous term, muscle tissue, there are three types of tissues, and maybe at the time we created this table to compare them. So there are some differences and there are some similarities between them. Cardiac muscle, like smooth muscle, is a muscle under involuntary control. You do not need to have conscious decisions of contracting that muscle for it to contract. This is going to be taken care of by the autonomic nervous system. The autonomic nervous system will send the signals to stimulate the cells. Well, actually, there are pacemaker cells that are just inherently contracting, and autonomic nervous system is just going to regulate this contraction. The cells are striated. The striation is present also in skeletal muscle cells, but not in smooth. And you can see the striations under the microscope as this basically zebra pattern of light bands and dark bands and light bands and dark bands within the cell. And between the cells, you have what's called intercalated discs. So these thicker lines here are the edges of the cell, basically, at what we call intercalated discs. Cardiac cells are attached to each other via these very specialized junctions, intercalated discs, that are rich in gap junctions. So intercalated discs, what they are gap junctions between the cells, and we've seen gap junctions between two osteocytes. We've seen gap junctions in just in the beginning when we first talked about cellular junctions. Gap junctions are basically membrane proteins that form tunnels between cells to allow the exchange of content between the cells. So there's exchange between the cells via these gap junctions. And this is going to be very important for the exchange of ions between the cells. And so the action potential can propagate across the cells and all the cells can contract. As what I didn't say about the striations before I jumped into the intercalated discs is that the striations is a visual of the organization of the contractile proteins inside the cell. So you can visually see them at the naked eye. These contractile proteins are organized in such a way, so parallel to each other and very organized, and we'll get into that organization, that you end up seeing a pattern that is visible under the microscope, because some of these contractile proteins are thicker than the others, and the banding pattern ends up being striated and repetitive because it's very, very organized. So these contractile proteins organized in this fashion are visible in the cardiac and the skeletal muscle cells. That doesn't mean they're absent in the smooth muscle cells. That just means they're not as organized, but they're there because smooth muscle cells contract as well. One nucleus per cell, just like smooth muscle cells. Skeletal muscle cells are going to have multiple nuclei. So let's look at smooth muscle. And, obviously cardiac muscle, I didn't mention where it's found, it's the heart. Smooth muscle is non-striated, but it has contractile proteins, but you just do not see it under the microscope. So visually, you can't see them organized the same way as they are in cardiac and skeletal. And smooth muscle, just like cardiac, is involuntary. This is the type of muscle you find around the alimentary canal. As it contracts, it moves the food down the esophagus, down the intestines, and it's also around the stomach to help in that churning process of liquefying the food. Smooth muscles are around the bladder. So they're throughout our body. They're around blood vessels to constrict blood vessels, or when they relax, it dilates the blood vessel and allows them to be wider and more open. They're around bronchioles in the lungs, and if they contract, then the bronchioles kind of like close up, and there's more difficulty breathing. So they really are controlling a lot of passageways for air, food, blood. Yes. So there is also just one nucleus for every cell. The cells are much more fusiform shaped. See, these are just two cells next to each other that are very, very fusiform, we say, or fibroblastic shape. A shape that actually I didn't discuss in cardiac. These are very rectangular shaped cells. It's kind of very obvious here. And some of them are actually split. They're kind of branched. Maybe not this one, but sometimes they're kind of like branched like this. But most of them are just rectangular with a central nucleus. So in smooth muscles, the cells are much flatter. There's a diagram here I included to show you also, in the walls of vessels, we have different layers of smooth muscles. It's in the tunica media of vessels. And this tunica media is a little bit, different thicknesses depending if it's an artery or a vein. but that's where you find smooth muscles around vessels. Yes. All right. Speaking of skeletal muscles, they're striated, and you can see this is like an amazing a dark band, light band, dark band, light band, dark band, light band. These are the contractile proteins that are laying down in the cell, and they're just very organized. So they end up creating this very geometrical pattern of parallel lines. And it is so full, it is so packed of contractile proteins that the nuclei are shoved to the edge of the cell, and they're kind of squeezed underneath the cytoplasmic membrane, right? So this is the cytoplasmic membrane of this cell, and this is the cytoplasmic membrane of the cell above it. There's the nucleus. So these are multinucleated cells. We only kind of see one nucleus for this cell and one nucleus for this cell. So unfortunately, maybe this nucleus belongs to this cell, so you can see two nuclei for this particular cell. But really, they are multinucleated. There's multiple nuclei within each cell, and the cells are very, very long. Cardiac were kind of short and rectangular. These are very, very long cells. You don't see the beginning and the end of these cells. They're very, very long. And skeletal muscle is under voluntary control. You have to make a conscious decision to move your legs, to walk to the door, to raise your arm. Okay, speaking of an organ made of muscle, you need to usually, we always see a connective tissue wrapping. We saw this around bone. We saw this around cartilage. We see this around brain, right? The meninges around the brain, periosteum around the bone. We have connective tissue wrapping around muscle. Each individual fiber, muscle fiber, which is also called myofiber, is actually one cell, one muscle cell. And it's just very, very long, and that's why we use the word fiber. Fiber is always referring to something that's very stringy, long. Collagen fibers, and this is just one protein. Muscle fiber, this is an entire cell, so it's completely two different fibers. So there's not one kind of fiber. The word fiber is really almost like an adjective to describe the actual structure of what we're talking about, so whether it's just one protein molecule or it's an entire cell. So muscle cells are very, very, very long and thin, and each one is wrapped with endomysium. This is the wrapping around each individual cell. So the cell is in, it looks like this dark pink here, and then you have the white halo. This would be one fiber. Yes, you can see it here. One fiber. So around each muscle fiber, you have an endomysium. And then the fibers are kind of bundled into kind of mini bundles within the entire muscle. And these bundles are called fascicles. So the bundling of the fascicles is made with perimysium. So perimysium is the connective tissue that is around a fascicle. So to wrap the myofibers, we use some perimysium, and we created a bundle that we call fascicle. And then several fascicles, so there's one here, another one here, another one here. All of these fascicles are going to be wrapped on the surface with epimysium. This connective tissue, so this is connective tissue, endomysium is connective tissue, perimysium. This connective tissue is going to blend with tendons, which is another connective tissue that is made of dense, regular connective tissue, we call it, because it's made of collagen fibers that are parallel to each other and anchor into the bone. More specifically, they anchor through the periosteum of the bone. So that would have been even more accurate if they had drawn a periosteum over here. All right, beautiful, well, a bit fuzzy here. A picture of cross-section of a muscle with endomysium around each fiber. So this is a fiber, this is another fiber, another fiber, another fiber. So because they're more like circles here, they're coming at you, right? You see these circles, right? These are the individual fibers. And around each fiber, the thin blue line is the endomysium. And then you have perimysium, a little bit thicker here, perimysium that bundles a fascicle. I can't really tell where's the edge of that fascicle on the other end here. But we have, it looks like there's a fascicle over here. We have several fascicles that make up this muscle. And then you have epimysium on top. Okay, so I added the diagram here. That's basically the equivalent of what almost the equivalent of what I used 2 slides ago for you to practice labeling the connective tissue layers. Okay, and this is one diagram from a different source. And I always forget to mention my source. I literally pull these pictures from so many different sources. It's hard to go back and find out where did I get this one from. But I love this one. I love this diagram because it's really like a 3D rendition of these of what's going on inside the cell. All right, so let's talk about this. This is one muscle fiber right here. You're looking at one muscle fiber. All of this is just one muscle fiber. So these individual tubes here, I'm going to call them, this is not the same as what you see here. This right here is one muscle fiber, right? This here, this one is one muscle fiber, is one cell. In this picture, the entire thing is the one cell, and now we're looking inside of it. Inside of the cell, we have myofibrils. Don't confuse them with the cell themselves. But they're run in the length of the cell, and the cell is very, very long. And inside this very long cell, you have all of these very, very long tubes that are literally next to each other and running parallel to each other. These tubes are made of contractile proteins. Myofibrils are contractile proteins that are all bunched together. They're all bunched together, and we're gonna, get into the nitty-gritty of myofibrils in a second. But I wanted to show you the rough endoplasmic reticulum. In a muscle fiber, it's called the sarcoplasmic reticulum. Anything sarco is referring to muscle, skeletal muscle. So sarcoplasmic reticulum is the equivalent of the ER, the endoplasmic reticulum in any regular general eukaryotic cell. Sarcolemma is the equivalent of plasma membrane. What else is sarco? Well, we're going to talk about sarcomere. within these myofibrils. So anyway, so these myofibrils, they are wrapped with this sarcoplasmic reticulum, and this picture really shows that very well. That's why I like to use it. This wrapping, it looks like a fishnet that's all throughout, wrapping each myofibrils. And we're going to see how important this sarcoplasmic reticulum is, because it's going to be housing calcium ions inside the terminal cisternae. Inside the sarcoplasmic reticulum. So the sarcoplasmic reticulum is in blue here. All the blue is sarcoplasmic reticulum, and that's where the calcium ions are stored. In pink is the terminal cisternae. So what are terminal cisternae? You see this? This is the terminal cisternae. You see that hole over here? That hole, or that mini tube that you see, is perpendicular to the sarcoplasmic reticulum and shoots up to the holes that are on the surface of the sarcolemma. So the muscle cell is riddled with holes that basically, these holes are actually invagination of the membrane. It's the membrane that goes down into kind of like a tube inward, and this tube is connected to a T tubule. It's kind of like a network from the plasma membrane coming down, and it just creates this network underneath the membrane. So this hole, these holes, it goes down the T tubule in here. And we're going to see what's the role of these two tubule in contraction. Mitochondria are all throughout the myofibrils. You can see them here. And of course, they're a powerhouse inside cells. They're the ones that produce ATP, and ATP is going to be much needed in contraction, in muscle contraction. So we need the ATP. It comes from the mitochondria, and this is the proximity of the mitochondria. So the T tubules, they happen to fall right between sarcoplasmic reticulum sections. And the end of a sarcoplasmic reticulum is called the terminal cisterna. And the end of the next sarcoplasmic reticulum section is called terminal cisterna as well. So you end up having this really cool triad where you have one T tubule and two terminal cisternae, and this is your triad. And so I'm presenting you the structures, and then when I talk about muscle contraction and I say triad, you'll know what I'm talking about. All right, so you have this fishnet, which is a sarcoplasmic reticulum around these myofibrils, and the T tubules are actually connected with the surface of the sarcolemma. So the T tubule, basically, there's this connection between the outer surface and the inside of the cell, and it's in proximity, right next to the terminal cisternae of these different sarcoplasmic reticulum sections. Okay, I spent some time on this because students confuse myofibrils inside the cell with muscle fiber, which is the cell. And then all these muscle fibers are in a fascicle like that, like this. These are muscle fibers in a fascicle. They think that's what this is, a muscle fiber in a fascicle. No, this is myofibrils inside one myofiber. That's what muscle fiber is also called. All right. So I really wanted to include as many flow charts I could, just to really make it clear the difference between all of these terms. So we went from bone, periosteum, around the bone, tendon, fascia, epimecium, perimecium. Now we're down into the endomecium around each individual cell. And then one cell is represented here. This is one cell. One cell. Yes. I'm just trying to like make sure. Right. So this is the fascicle. All of this is the fascicle. And then you have each cells in a fascicle. So this is one cell. And inside the cell, you have myofibrils. And they're, all throughout the cell. So here there's, whatever, a dozen here. Okay, and then what makes up the myofibrils are the contractile proteins or filaments. Thick and thin filaments. So let's look at that. What makes up a myofibril? These rods that I showed you 2 slides ago, right? These rods here, what makes up these myofibrils? Because that's basically the main component of muscle cells. So what is that? So that's a combination of thick and thin filaments. Thick and thin filaments make up the myofibrils. See, we're going deeper and deeper into the cell. So we're really zooming in now on thick and thin filaments. So the thick filaments, they're called thick filaments of myosin. And as you can see, we have actually 2 polypeptides to make up one myosin molecule. 2 polypeptides that are intertwined with each other. So they form they can form one molecule. It's kind of like the hemoglobin is made of 4 polypeptides that are come together to create this macro molecule called hemoglobin. So 4, globular peptides. Here, these are filamentous polypeptide because they're long, or we can say fibrous polypeptides because they're long. And the 2 of them, and they wrap around each other to form what's called a tail. And at one of their ends, there's actually an expansion, and that's called the heads. So the 2 heads are next to each other and the tails are intertwined. And we really don't talk about these 2 polypeptides anymore after they're originally presented. We just draw a thick filament as one tail, one head. That's basically how from now on I'll be drawing it. But technically speaking, the whole thick filament is made of 2 polypeptides intertwined with each other. Okay. These myosin filaments, thick filaments of myosin, are laying next to each other, faced by their tails. So I give the analogy of a slumber party, and everybody's in their sleeping bags. And there's just really, it's a very tight space, and we have a lot of friends here coming over. And we just kind of like, put a number of them on one wall and the others on the other wall, and everybody's facing each other by their feet, and everybody's in their sleeping bag. So the heads are the heads of all of these friends, and the tails are basically their legs kind of laying next to each other here in this slumber party. So we end up with a region that's kind of bare of heads, and two opposite regions that is rich in heads, but they're opposite regions. And the way that they are assembled, like next to each other, laying down next to each other, they form this tube. So this is actually circular. It's a tube, not a hollow tube. It's a solid tube of all of these thick filaments facing each other by their tails and their heads on opposite ends. Like this. So you have, right? So this is like a thick middle portion, and all the heads are sticking out. Okay. Thin filaments, so I'm describing them separately, and then I'll show you how they're interacting with each other. Thin filaments are they're made of actin. Thin filaments of actin. So we're just looking at those little beads here, these red beads. We're not looking at the white lines and the yellow molecule here. I'm going to talk about this next. So all of these yellow beads, so the thin filaments of actin, they're actually made of globular actin. We say G actin. This is one G actin right here. One G actin, and this is another G actin, and another one, and another one, and another one. So plus one G actin plus etc., creates one long F actin. F actin. F for filamentous, because actually we end up with a very long molecule. The individual monomers are globular, and when they assemble together, they form this long filament. So it's like a pearl necklace that is kind of not closed, but just kind of open and laid flat, and you have all of these pearls of G actin, and they form F actin. The thin filament is actually made of two of these strands. So F actin is basically one polypeptide. And you need two polypeptides to form a thin filament. So two of these F actins to form a thin filaments. You see how I built this up? G actins are individual monomers, and they kind of string together to form this long F actin, and you actually need two F actins together to form the actual thin filaments. So just like the thick filaments, the thin filaments is made of two polypeptides that are kind of intertwined with each other. So when we draw it, I'm going to use this thicker pen here. And this is kind of what we draw. We do this, and maybe a slightly different color here. Like this, okay? So these two polypeptides of F actin are kind of intertwined with each other to form the thin filament. Okay, now let me show you, let's see. Well, okay, I'm gonna talk about this next. Let's talk about the regulatory proteins. So thick filaments of myosin and thin filaments of actin are your two contractile proteins. That's it. Those are the two major players. Myosin and actin, thick and thin filaments. Now, we also have additionally on the surface of actin, not myosin, on the surface of actin, we have regulatory proteins. that are called troponin and tropomyosin. So these regulatory proteins are sitting they're sitting in the grooves that are formed by this intertwining. So let's go back here. You see this intertwining that I drew. So you would have one here, one here, one here, right? So whenever there's this groove, you end up having this long tropomyosin. This is tropomyosin with a big T. Tropomyosin right here in the groove, all right? And it happens to basically cover 1, 2, 3, 4, 5, 6, 7, on average 7 G proteins. So tropomyosin, I'm just going to draw a big T, tropomyosin, covers 7 G proteins. So there's one G protein, another one, another one, another one, another one, another one, G-actin, G-actin. So these G-actins that are strung together and part of the longer F-actin, they're actually covered by the tropomyosin. So every 7 gets a new tropomyosin, and the next 7, another tropomyosin covers them, etc. And then we have a smaller molecule called troponin. Troponin binds tropomyosin. Here's troponin. Troponin is right here. And what troponin is a calcium binding protein. Very, very important. Troponin is the calcium binding protein, and learn that. It's not tropomyosin, it's troponin. Troponin is calcium binding protein. So I give the analogy of, yeah, we're gonna see that. I give the analogy of troponin, tropomyosin being like a, yeah, I'm gonna actually draw it. Perfect. This is tropomyosin in light blue, light green, and then this is troponin in dark green, and calcium. Let's make calcium in black. There we go. So calcium in black here. So this is calcium. My analogy is calcium is a key, and that fits only into one lock, which is on the doorknob called troponin. Troponin is the knob that has the keyhole for calcium. And once calcium binds on troponin, the whole complex, troponin, tropomyosin, because tropomyosin is the door, by the way, I think you guys said that. The whole complex, troponin, tropomyosin, is going to swivel out of the way. So here are the hinges, and when calcium binds on troponin, you have the entire door that swivels open. That swivels open. Maybe I should have done it this way. There we go. Perfect. So calcium binds on troponin, tropomyosin, troponin complex swivels out of the way. So when we say swivels, it literally swivels. This complex right here is going to go just like that. It's going to swivel out of the way. The whole complex, so it's like a door on its hinges, it's going to swivel out of the way. It doesn't fly away, it doesn't disappear, disintegrate, it doesn't collapse. It just, in biochemistry, we talk about bioconformational change. The molecule changes conformation, means it changes shape, literally, and it's going to kind of like bend out of the way. Okay, so why is this kind of like cool? Because these little, I'm going to use red, these little individual G proteins in red, these G proteins, they actually have a binding site on them. Each one has a binding site. Like this. And they're strung together and they form F-actin, right? So each one here actually has a binding site. Binding site. A binding site for what? A binding site for the heads of the myosins. Boom. Boom. So I'm going to explain to you when the binding takes place, but I want to kind of show you all the players that are involved, what their structures are, so you can understand how they interact with each other and create the movement that we're going to talk about. So each individual G actins has a binding site on its surface that is specifically for the heads of the myosin. Okay, so where is myosin in my drawing here? I don't even see it. That's because this drawing is just about the actin thin filaments with these regulatory proteins on top of it. We're going to see now everything together. All right, so now we're overlapping thick filaments, thin filaments. How are they laid out together? Just like this. So you have the thick filaments that are facing each other. Remember my slumber party, they're facing each other by their tails, and you have all of these molecules kind of laying down like this, etc. And then more on this side. Etc. And it continues, right? So we have, more over here and over here. Oh, you're not giving me enough space. Just kind of sleep over here. Anyway, so we have this bare zone. It's bare of heads. There's no heads. And then we have these ends, the two ends, that are very rich in these myosin heads, and literally in a kind of like a circle fashion, because it's a rod. It's an entire rod. Where are the thin filaments? Right over the heads. Boom. Hi, here we are. We're here to maybe bind together and hook up. So these thin filaments, look at that. They are right here over the heads. And these thin filaments, they're made of individual G actin that assemble into F actin, right? So these individual G actin they each have a binding site for these heads. So you can see the thin filaments are above, the heads are below. Hey, you can imagine they're going to hook up that way. So that's the pattern that we see as a striation under the microscope. We have very, very thick filaments that are laying, parallel to each other. And then we have thin filaments that are also laying parallel to the thick filaments and parallel to the other thin filaments. Everybody's parallel, but just over the heads, which means there's an area where you only have thick filaments and an area where you have thick and thin filaments. So here, only thick. I have a pet peeve with when my pen lags. It just bothers me so much. Okay, and an area where you have both thick and thin. Thick and thin together. So now it's that these areas where both thick and thin are together, it's just more material for the light to get through. So the light microscope, the light that's coming out of the bulb in a microscope, it goes through the tissue. And if the tissue is thicker, it's going to be, it's not going to pass through as much as if the tissue is thinner. So when we have areas of just thick filaments versus areas of both thick and thin filaments, we're going to see more light coming through the bare zone than light coming through the other parts where you have both types of filaments. So you have dark areas and light areas, and again, dark areas. And you can see, wow, this is going to create dark, light, dark, light, dark, light. And it looks like a striations of like a zebra pattern, we say. But, you know, it's just due to the fact of how these parallel fibers are laying down, right? They're all laying down horizontally, but because of their overlap, you see this kind of dark light, zebra pattern. Okay. Throughout these filaments, you have also elastic fibers that are, let me just kind of highlight them. These elastic filaments or fibers, they're there to basically anchor the thick and thin filament bundles. You see, this is what thick and thin filaments are. It's illustrated over here. This, with the bare zone in the middle. Remember the bare zone that only has the thick filaments, and then thick and thin are on both ends over there. So you have this entire section of thick and thin filaments, and then you have elastic fibers or elastic filaments that are anchoring these contractile filaments in the middle to Z-discs. Boom, 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 boom. What the heck are Z-discs? Z-discs are proteins that are just arranged more vertically and act as like a wall to anchor these fibers on. That way we have kind of an anchor point, and right there, and it doesn't span the entire cell. We have it just there. And the span between a Z-disc and the other Z-disc, that's what we call the sarcomere. So I want to write it, but it's lagging. Sarcomere. Okay, so sarcomere is the unit of contraction, but it's just that. And then there's another one next to it, and another one next to it, and another next to it. So now basically what we're doing is we're zooming back out, right? We went very deep and looked at the molecules and how they're kind of like laying parallel to each other. And then we're zooming back out to see, okay, at the end of this arrangement, this bundle of rods of thick filaments with their thin filaments over them, that is anchored to the walls of the Z-discs by elastic filaments. And then you have another bundle next to it over there, and it creates another sarcomere next to it, and next to it, and next to it. And all of these sarcomeres are next to each other and anchored on the edges of the cell. So when they contract, they're all going to be pulling inwards, and the whole cell is going to shrink. It's amazing. So here's the sarcomere. You should know the sarcomere. And I'm really hoping all this is really a review for you. And maybe you want to skip the video and skip to the parts that might be less familiar to you. But I'm explaining it just in case. So here is a Z-disc. And here's another Z-disc. And here you have the H band. This is the sarcomere. The area between from the edges, the two ends of the thick filament is called the A band. for anisotropic. Actually, it's going to come up in the next slide. And then the area where you only have thin filaments is called the I band. So the thin filaments, which are illustrated in orange here, the thin filaments are on, on both sides of the Z disc. You have thin filaments because, for example, these thin filaments, they are part of the next sarcomere. And these thin filaments are part of this central sarcomere. So the I band is actually two halves. There's a half of an I band in one sarcomere and the other half of the I band in the next sarcomere. The whole I band actually kind of spans two sarcomeres and is bisected by the Z disc. Okay, so a sarcomere is basically half of an I band, an entire A band, and half of the next I band. Right, so I bands, we have half on one side, half on the other, and the A band in the middle. The H band was the bare zone where there was no overlap between thick and thin filaments. All right, so that's the sarcomere. So as I said, you have these alternating fibers, and in some areas you have both thick and thin, and other areas you only have thick filaments. So that really has an impact on the light going through the tissue. Dark A bands are A for anisotropic, and I is for isotropic, and that's for the way that the thickness affects that polarized light coming from the microscope. All right, so and I said all this already verbally. So this is an actual electron microscope. You would not be able to see this with a light microscope, a regular light microscope. This is an electron microscope. And you see here the nucleus of the cell. I mean, we're really, really, really close. Like we are so close, you're kind of almost inside the nucleus. And what you see is this banding pattern. And of course, in the middle of an I band, there is this darker line. It's actually the Z disc. So you see this? The Z disc is in the middle of this I band. So in an actual picture, you kind of see this line. So the line is actually made of these Z proteins that form this Z wall or disc where we have anchoring. So when muscle cells contract, the sarcomeres shorten, and when the sarcomeres shorten, they pull on the Z discs inwards, and that eventually pulls on the sarcolemma, which is a plasma membrane, and the whole cell shrinks and becomes shorter. And when it contracts, it shortens. That's my song. When it contracts, it shortens, and when it shortens, it pulls on what it's attached to. No, it's definitely not a rhyming song, but that's what I keep saying. When it contracts, it shortens, and when it shortens, it automatically pulls on what it's attached to, and there's movement. Okay, now, do these filaments kind of like disintegrate and rebuild? No. Actually, these filaments do not change length at all. So how do we have this entire sarcomere being narrower? Because that's what's going on here. We have a narrowing of the sarcomere inwards. All the sarcomeres kind of get narrower inwards, and it basically overall pulls the whole cell shorter. It shrinks it. Without the filaments changing length. How is that possible? That's possible because they overlap each other already. And what happens during contraction, they're going to overlap even more. And that overlapping allows for a shortening, right? So if my elbows were the ends of the sarcomere, the movement of these filaments over each other make it that they've never changed, right? I never cut off my forearms, but my elbows are actually closer to each other. So these the ends of the sarcomere, they kind of come closer to each other without touching the filaments. So the filaments, all of this, how is the sarcolemma being moved? I mean, this is the filaments dancing inside the cell. How is it that the whole cell actually shrinks? It's because these filaments that are sliding and shrinking inside, the sarcomeres are narrowing, all of this is inside. Because at one point on the edge of this whole thing, we have molecules that attach all of these contractile proteins to the edge of the cell. That way, the whole cell shrinks with the sarcomeres. And these cells are basically linking cells that are linking the thin and thick filaments right here to the membrane. This is the membrane. The sarcolemma is the plasma membrane, the phospholipid bilayer. And let's look at these molecules. Dystrophin is one of them. And we have other linking proteins that the names are not mentioned here because I'm going to focus on dystrophin. But of course, you know, in a course that would be even more heavy on molecular biology of all of this, we would have all the names of these linking proteins. And it's research that allows us to identify these proteins and figure out that they're transmembrane or they only, you know, below the membrane or on the surface. I mean, it's really amazing. And there's so much that we still don't know, but there's constant research about all of this. So dystrophin is a linking protein. It's a linking protein that links all these contractile fibers, these proteins, to the surface. And why I'm talking about this? Because I want to make a clinical correlation. Muscular dystrophy is a perfect example of one molecule not working properly and creating havoc on the muscular system. It's kind of like hemoglobin in sickle cell anemia. One molecule. So this is a hereditary disease, so it's genetic that is passed on. That's what this means, hereditary. It's a genetic mutation that is passed on to, you know, offsprings. And the mutation is about dystrophin. And dystrophin is not produced anymore. So you do not have a connection between the contractile proteins inside the cell and the surface of the cell. So you have dystrophin, instead of maybe being widely distributed and you have an even shrinkage of the cell, you have only certain dystrophin produced, or there's I actually don't know the detail of if it's a malformation of the molecule itself, or do you just have less of the molecule? So when you have, I'm thinking if you have less of the molecule, then you have these torn cell membranes because you do not have a uniform linkage of the entire contractile system to the entire membrane. So when they're contracting, they're kind of pulling more on one side than the other, and then the cell tears. I'm assuming that it's a matter of abundance or lack of dystrophin that leads to torn cell membranes. So not a misshape. Okay, so a normal allele makes dystrophin, but when you have a mutation, the allele then is not normal, and it's passed down. Oops, we went really fast here. So there are several forms of muscular dystrophy. Duchenne and Becker types affect the shoulder, the arms, the thighs. Other forms, Emery-Dreifuss type, affects more the pectoral region and the arms, right? There's different forms of muscular dystrophy that basically affect different regions of the body, depending on where the mutation is being expressed. So in a genetic course, you would get more into this discussion of where the genetic mutation ends up being expressed. But here we're talking about one allele that is normal for dystrophin. So I would put a big D for normal dystrophin, and then you have a mutated allele. Oops. For the abnormal mutated form of dystrophin. So let's look at this genetics. So it's a recessive trait. That means if you end up with having at least one normal allele, you're okay. The absence of the expression from the abnormal allele is going to be basically compensated by the normal allele. So the normal allele will produce, oopsie, the normal allele will produce dystrophin. Yeah, this bothers me so much. Okay. The normal allele produce dystrophin, so you have some dystrophin, but if you inherit, so you're normal, but if you inherit both mutated forms, if both of your parents are carrier of a mutated allele, and you happen to inherit the allele that was mutated in your parents, both of the mutated alleles, that's very unlucky, and you have no dystrophin being produced. No dystrophin being produced. And that's when you're affected by the disorder. Right? So here you have a unaffected carrier. Oh, I forgot to mention one more thing. It's an X-linked disorder. Oh, this is very interesting. I forgot to mention that. So these two chromosomes that I just drew general chromosomes, but I should have said these two are X chromosomes, right? So the ones I drew here were X chromosomes. So if you're a female and you inherit one normal allele, it compensates for the abnormal allele. So here's the genetic tree of an unaffected father that marries a woman that actually is a carrier of the mutation. So she's unaffected. She's unaffected because the normal allele is compensating for the mutation. Okay, but now the problem is, if you have a boy and he happens to receive, out of the two X's that the mother could have given him, by random chance, the mutated chromosome X ends up being the fertilized oocytes by the sperm and ends up, giving you a baby boy, but with a mutated allele. This will be an affected son, because the Y chromosome, actually, this is unfortunate that they drew a Y that's that tall, because the Y chromosome does not look anything like the X chromosome. Carries genes, set of genes that are different. It's a really different chromosome. It's paired with the X chromosome as a 23rd pair because it's the sex pair, and okay. But in males, X and Y chromosomes are different. There is no compensation from the X chromosome. Oops. So the affected chromosome here is in pink, right? So this is the affected chromosome, so the dystrophin that with a mutation. And there is no, this is kind of, I'm just noticing this is a little bit misleading here. There is no dystrophin gene on the Y chromosome, and that's why you do not have a compensation, and you end up with just one gene for dystrophin that happens to be the mutated one, so you're not making dystrophin, and that's why this boy is affected. The girl has the other X chromosome, and both X's, they're the same, and they just carry the same set of genes, so dystrophin gene is on the other X, and it happens to be normal, so it compensates for that mutated one. Okay. Yeah, that's maybe I'll redraw this with my own little people and a set of chromosome that's not as misleading as this XY. It really should be, it should be something like this is X and this is Y. With the dystrophin gene being here. And there's no equivalent dystrophin gene on the Y chromosome. All right. In all my years of teaching this, I never encountered anyone with muscular dystrophy until a couple months ago. I had a friend come over, and her son has muscular dystrophy. He came in a wheelchair, and it's just devastating to actually see a person, a friend, with a son, and their life expectancy is limited. It's just really weird to actually see a patient with it. Yeah, so I added this particular slide from Netter to give you a bit more idea on the development of the disease. That son is the boy I met. He was my son's age, 14. He was 14. He didn't look as deformed, but he was wheelchair bound. All right, let's talk now that we talked about muscle contraction, let's talk about the neuromuscular junction. The neuromuscular junction is the junction between the neuron and the muscle cell. This is where the signaling is going to arrive. This is when the cell is going to be stimulated to contract. The skeletal muscle cells need a motor neuron to come and tell them, do it. So here are a couple of examples of motor neurons sitting in the ventral horn of the spinal cord, and they project their axon to their target cells, and each motor neuron actually splits into several terminal fibers, axonal fibers, and each axonal fiber reaches a different cell. So one neuron can actually stimulate several muscle cells at the same time, and another neuron will stimulate another group of cells at the same time, and another neuron will do yet another third group at the same time. This is done so not all the cells are stimulated by the same neuron, but they kind of take turn being stimulated. It just helps with the process of stimulating the muscle. In the microscope, you literally see the synaptic knob at the end of the axon. So this is the motor nerve fiber. When we say fiber, remember, this is making me upset. I'm going to have to figure out what's going on. The fiber is basically the axon. Axon. We see that's another example of a structure we call fiber because it's so long. So the axon of a neuron ends up in this kind of dilated end, and that's what we call the synaptic knob. And you see it here on the surface of a muscle fiber. So right here, the neuromuscular junction illustrated here. This is your axon. And this is that dilation I told you about, the synaptic knob. What they call it here? Did they give a different name? They just called it axon terminal. Okay. Synaptic vesicles are floating inside the synaptic knob, and they are packed with neurotransmitters. Acetylcholine is going to be the neurotransmitter that's going to stimulate the muscle cell. On the muscle side, we have, this is the sarcolemma. We already saw that term. And in the sarcolemma, we actually, it doesn't go straight down like this and, forming just these two parallel lines facing each other. Actually, we have these invagination of the membrane like this. This is called the end plate. And I don't see it written here. This is the end plate. The end plate is very rich in acetylcholine receptors. So when acetylcholine is going to be released from the cleft, there's going to be acetylcholine receptors binding, allowing for the binding of the acetylcholine. So very, very receptive area, right? The more receptors, the more we can actually respond to a stimulation. And look what's below that. All of these contractile fibers. So there's a lot of drawings coming up about contraction, but I'm having difficulties with the lagging of my pen. So I'm going to pause this recording, fix it, and hopefully get back to you. Okay, let's discuss the membrane potential. The membrane potential is a difference in charges on the surface of the cytoplasmic membrane of cells, and it's very important that it's established, especially in neurons and muscle cells, because that's the way to excite the cell. The excitability of the cell is a fundamental property of these two cells, neurons and muscles, and to be excitable, you have to have a membrane potential. So I'm going to draw the membrane, and here is the sodium-potassium pump, which is the main contributor of the membrane potential. There are other contributing factors to establish this potential, but this is one big one. So the sodium-potassium pump is an ATPase that pumps ions against their gradients. So ions are normally, they normally diffuse down their concentration gradient to reach equilibrium. This is kind of the natural thing that's going to happen for any molecule, any ions. If they're allowed to move through a membrane, they will just diffuse down their concentration gradient. So this is a diffusion process. If sodium ions were highly concentrated outside of the cell, less concentrated inside of the cell, there's a diffusion process that's going to bring them from the high to low. This is the definition of passive transport, and that's what's going to happen. The pump has the job of pumping these ions against their gradient. This is not going to happen naturally because they're already very concentrated outside of the cell, right? This is outside. And it's difficult to basically accumulate more ions and more ions. Think of it as your class is taking place in a room that only holds 20 people, and there are 80 students registered for this class, and everybody has to sit on everybody's lap. Nobody's going to be happy about that. And it's, you try to add 20 more students in the classroom and it's impossible because you can't fit them in. So that's the idea, is it's not natural for the ions and molecules to move to a compartment where they're already very concentrated. The idea is that they're going to distribute so there is an equilibrium between the two sides. The pump pushes them against that gradient and uses the energy of the breakdown of ATP to do that. Breaking down ATP releases energy, and the energy pushes these ions against their gradient, So this pump is called the sodium potassium pump, because it's going to pump both of these ions against their gradient in opposite direction. So we have, for potassium ions, the pump keeps them highly concentrated inside of the cell when, and less concentrated outside of the cell. So by diffusion, these potassium ions are going to have a tendency to diffuse down their concentration gradient, passive transport all the time, and the pump keeps pumping them back against their gradient, to keep this this distribution. Now, it does this unevenly. For every 3 sodium ions it pumps out, it really only brings 2 potassium ions back in. So in the end, 3 plus charges are pumped out, but only 2 plus charges are brought in. 2 positive charges in, 3 positive charges out. Now you are also messing up with the difference in charges across the membrane, not only the difference in the concentration of each individual ions, but overall the charges are going to be more positive charges on the outside than on the inside. Additionally, inside the cell, there are just more proteins, and proteins are negatively charged. DNA is negatively charged. There's a lot of negative charge on the surface. So in general, the inside of the cell is more negatively charged in general, and this just adds to this. So at the membrane, this is the membrane, we have a potential. The word potential means a difference in charges. Difference in charges. And that's how the cell likes it. That's how the cell needs to be if it has to be responsive to a stimulation. Because the way we can wake a cell up and stimulate it is if we tweak these charges and we mess up with this potential, then the cell realizes, oh, what is going on? Something's going on at my membrane. It's a stimulation. If a depolarization occurs, it's the signaling of a stimulation. So we're going to talk about depolarization. So that's the membrane potential. So it's very, very simple, very straightforward. Now, how do we excite a muscle cell? And you can use these pages to redraw this on your own without looking, after you kind of take notes from reading or hearing this video. The excitation phase, so now there's going to be 4 phases to muscle contraction. The first phase is excitation. First, we need to excite the cell. If we can't excite the cell, nothing's going to happen downstream. And then we're going to have the excitation contraction coupling. How do we get the excitation from the surface to the contractile proteins inside the cell and let them know that there is a stimulation on the surface? Then comes contraction, then comes relaxation. So let's talk about excitation. So the excitation takes place when you have the axon terminal reaching a sarcolemma. So the sarcolemma over here in red. So this is the sarcolemma. And this is the axon terminal. This is the neuron, motor neuron, coming here to stimulate this particular muscle cell for contraction. So there is an electrical impulse that we call the action potential. The action potential is in the form of a depolarization at the membrane. So when there is an electrical impulse traveling down the membrane of a neuron, the idea is we need to transfer that electrical impulse to this muscle cell. And once it's transferred to this muscle cell, then we have an excitation. So the membrane potential is normally this, right? Negative on the inside, positive on the outside. And it's also the case in a neuron, right? Negative on the inside, positive on the outside. But when the electrical signal comes, there is a depolarization that's running. That's what the action potential is. Actually, we talked about that in the nervous system, and I reviewed this with the synaptic transmission between two neurons. So when this action potential arrives here, because it's a difference in charges, a distribution across the membrane, we have voltage sensitive channels. I'm going to draw them in blue. Voltage sensitive channels that are for calcium. So this is the key here. These are going to be voltage. We also say gated, voltage gated channels in blue. Voltage gated channels, they open their gates when there is a voltage change at the membrane. If you tweak the voltage here at the membrane, these voltage sensitive channels pop open. This particular one at the synaptic knob at the end of the axon terminal happens to be for calcium ions, and it allows calcium ions to come in from the extracellular fluid. So calcium ions enter the cell, calcium concentration rises in the cell, and what that does is it promotes the exocytosis of synaptic vesicles that were just kind of sitting there and just kind of like bopping inside the synaptic knob. They're packed with a neurotransmitter, right? These are neurotransmitters. Well, one neurotransmitter in these vesicles. But when calcium levels rise, and it's a very common story in other physiological events in the body, levels of calcium rising in a cell promotes exocytosis. That's how it works. So the exocytosis here is going to be of the acetylcholine. That's the neurotransmitter in question here at the neuromuscular junction. That's what we're talking about. Okay, so acetylcholine end up in the synaptic cleft. That's great. What do we have on the opposite end? We have acetylcholine receptors. Acetylcholine receptors that happen to also be channels with a gate that's closed. And this is the receptor portion. And all of these receptors are kind of like waiting patiently, empty, right? They don't have acetylcholine on them and they're closed. The channel portion is closed, the receptor is empty, and they're very numerous in the end plate. Remember the end plate in the sarcolemma at the neuromuscular junction? We have this membrane that actually goes up and down right here in that region. And it's very rich in acetylcholine receptors. And these are ligand-gated channels. So in green here, ligand-gated channels. Okay, so ligand-gated channels, if you tweak the potential at the membrane, they're not going to care. This is not how these channels work. These channels are going to work once you have the binding of the ligand. And in this story, the ligand is acetylcholine. There we go, acetylcholine. So acetylcholine released in the synaptic cleft is going to bind on the receptor portion of this ligand-gated channel. And then the channel opens. Boom, the door opens. So this was the receptor portion, and this was the gate. The gate is closed here. And the gate opens. These gates are going to open, but only when the ligand binds. See that? Okay. The gate opens, so that's beautiful because these ligand-gated channels are for sodium ions. So I'm just going to draw two more here and draw the sodium ions. The sodium ions, yeah, just in black. Okay, I'll just keep them in black. The sodium ions, we learned from the membrane potential here that the sodium ions are in high concentration outside of the cell, right? This is a normal membrane potential. So when you open channels for these sodium ions and you basically kind of let them choose where they want to go and open the door, these sodium ions are going to have a tendency to go down their concentration gradient. That's the natural process of movement of ions down their concentration gradient. So you basically are bringing a bunch of positive charges inside the cell when you open these gates for sodium ions. So acetylcholine receptors are also channels for sodium ions. They are sensitive to the binding of acetylcholine, and the channel will open when the gate of the channel will open when the binding takes place. And here the ligand is acetylcholine, and the ion in question is sodium ions. All right. So if you put electrodes on the inside and the outside of the membrane of a cell, you would read a membrane potential that's around 70 millivolts. If you add positive charges inside the cell, you're adding positive charges, that means you're going to start tweaking that potential. It's going to affect the potential. Now, it's not going to be negative 70 millivolt. It's going to be less and less negative because you're bringing plus charges that are basically canceling some of these negative charges. So it goes from negative 70 to maybe negative 68 millivolts, negative 65 millivolts. The more channels open, the more we have depolarization. This is called a local depolarization. We also call it an end plate potential because this is the potential that is created at the end plate, this region here. This is the end plate. Okay, so this is a local depolarization. And if we have enough sodium ions that came in where we finally reach negative 55 millivolts, that's the threshold. The threshold, I'm not going to write the word threshold, but you should know it. The threshold is negative 55. And when negative 55 is reached, so this will depend on the number of channels that open and what kind of ion comes in. Are we bringing positive charges or are we bringing negative charges? That's going to go in the opposite direction. So once you have reached the threshold, now you have voltage sensitive channels that open that were kind of sitting downstream of the sarcolemma, and they were waiting. They were normally closed, so I'm not going to draw the gate closed, but they were closed. The channels were closed. These are voltage sensitive channels, so they're watching all over there, they're watching all of these ions coming in because of acetylcholine binding, and they're like, this is okay. I guess that's how you work. This is not how I work. I work if the membrane potential changes because I'm voltage sensitive. Okay, I'm changing the membrane potential. Why aren't you opening? Well, I really don't want to like bother myself until we reach negative 55. If we reach negative 55, well, that means the depolarization is strong enough. I'll join. And when these voltage sensitive channels, see that we reach negative 55, they finally open. The gate opens, and they happen to be voltage sensitive channels for sodium ions. So now we have even more sodium ions in. Boom. When voltage sensitive channels open for sodium ion and the sodium ions rush in even more, so then the depolarization is huge, and that's the firing of an action potential. And this, once these channels open, it's like a domino effect. Once you open them, there is no coming back. It's just like we talked about between two neurons. The action potential is fired. It's a non decremental event. It's just going to go all the way down the membrane. So it reaches even plus 35 millivolts here inside the cell because we brought in all of these positive charges. And then it will, repolarize when we allow potassium ions to exit the cell to repolarize the membrane. But I just don't have room to draw that, the repolarization of the membrane. But we discussed that when we talked about synaptic transmission between two neurons. Okay, that's the excitation phase, phase number 1. That's phase number 1, the excitation phase. And basically the goal was to recreate the action potential on the target cell, and we did just that without even touching the target cell per se. We just sent a messenger. The messenger locally created a small depolarization that was strong enough to then open the additional channels for a larger depolarization at the membrane. Great. The cell is excited. It knows it has to contract. So the next phase is called the excitation contraction coupling. So we should add the word coupling here. Phase. And this is the part where you need to take that that excitation on the surface of the cell and transfer it down inside to the contractile proteins to let them know that the cell got excited and we need to start moving, right? Moving the filament so we can shrink the cell and we can move this structure in question. So the structures that are going to be involved in this coupling process are these T tubules that I talked about earlier. So I'm going to draw the sarcolemma, and I'm going to draw a T tubule. And there's the sarcolemma. And just like we had in the previous diagram, so I'm just going to go back to it just so you see where I'm taking this from. Here, right? I'm drawing the T tubule. I picked the red color for the sarcolemma, and I'm picking blue now to show you the terminal cisternae of the SR. All right, that's what I'm drawing. But I'm drawing it as a side view and not a superior view looking down. Here is you're looking down at the whole structure. I'm drawing it as a side view. So here we are. So the SR in blue, the SR is on the sides here. So the SR is an organelle. in the cell, right, is the equivalent of the endoplasmic reticulum. So it's an enclosed organelle, and I'm just drawing the last portion of that compartment, which is called the terminal cisternae. And it's just a meshwork, and it's kind of harder to draw the whole meshwork. But what's really important is that inside the SR, right, this is the SR, and this is the T-tubule. And the SR contains all of these calcium ions. The SR is the reservoir of calcium ions. So calcium, calcium, calcium inside the SR. Okay, just the terminology here. This was terminal cisternae. And this is another terminal cisterna. Terminal cisterna, terminal cisterna with the T-tubule, and that's how you have your triad. Remember, 2 terminal cisternae, 1 T-tubule form the triad. Okay, so why is this setup important? Because we created an action potential on the surface of the sarcolemma. Remember, that's where we are here. The action potential is created, perfect. So now we're up here. How do we let the inside know that, you know, we have an excitation on the surface? We use the T-tubule. So this depolarization of the membrane, right, plus, right, this is a depolarization, which represents the action potential traveling down. And so imagine all the dominoes going, opening, opening, open, depolarization, depolarization, boom, 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 boom. And it literally, there's a depolarization that is created all along the membrane like that, one domino after the other. And then there is repolarization behind, where these dominoes are being put back up with the efflux of potassium ions, and it just regenerates that positive surface. But right here, we're just following the action potential. So the action potential, which is this big depolarization, is moving along the membrane. So you have these voltage gated channels all along the membrane, and they are opening, and depolarizing the membrane, depolarizing the membrane. So what happens is that there are voltage sensitive channels on the terminal cisternae, and the voltage sensitive channels, I had drawn them in blue. So these voltage sensitive channels, here they are. They are sensitive to a voltage change. Here's the action potential coming down the T-tubule. My analogy is like the action potential is taking the roller coaster ride down the T-tubule, and and all of these voltage gated channels are so excited. There's this depolarization, and they are sensitive to voltage change. So like poof, they open up, and those voltage sensitive channels happen to be for calcium ions. So calcium ions, because they're very concentrated inside the SR, they're going to have the natural tendency to escape, right? They're going to go from a compartment of high concentration to a compartment of lower concentration. All right, so the calcium ions go down their concentration gradient from this very concentrated compartment to outside where they are less concentrated. Actually, there's a pump that pumps them back in at the end of the whole muscle contraction. So they're very, very concentrated inside the SR. So when you open the doors, they're going to flood out and come outside. And guess who is very interested in the availability of all of these calcium ions? It is the troponin-tropomyosin complex. And I'm realizing I don't have room here to draw that. So I'm just going to add a page. Current template, blah, blah, blah. Here we go. So when you have all of these calcium ions available, wow, this is perfect. This is for the troponin, tropomyosin complexes that are covering the globular, now I forgot what color I used for that, but I guess that's okay, right? So these were the G proteins that had these binding sites. I think we had counted 7 of them, and they were all blocked. But when you have calcium that binds onto troponin, right? This is troponin, small t, and tropomyosin is the larger molecule, my door, my doorknob, and my key. And when the key is in the door, you end up being able to swivel the door away from these binding sites. So the binding sites end up being revealed. I'm kind of drawing them a bit lower here. They are revealed. They are free to bind the heads of the myosin. Okay, so that's what I wanted to add. So what happens next is contraction. Now that we have calcium available, now that we can actually reveal the binding sites on the heads on the actin monomers, and the heads of the myosin can bind on the actin, now we can actually have the binding of the 2 types of filaments and have their sliding over each other. And that's called the sliding filament theory. So myosin happens to have an ATPase property to it. The myosin had, not only is it a contractile protein involved in the contraction, but the head has an enzyme property to it that it actually can cleave an ATP to release the energy it needs. And that energy is going to be used to cock the heads of the myosins, which means to bring them out, flex them out, so they can bind on the G-actin. And form the famous cross bridge between the 2 types of filaments. So let me draw that. I'm going to actually draw these G-actin upside down. And I'm going to draw the heads of the myosin this way. And the ATPase allows the heads to cock, which means they're, extended. Did I say flexed earlier? I meant to say extended position. I think I said flex. It shouldn't have been said flex. Right, so these are extended positions like this. I'm going to draw a third one. So the heads go into an extended position and clip into the binding sites of each individual G-actin. So this is a G-actin. All right. And remember, these myosins are facing each other by their tails, and their heads are on the other side as well. And I'll just draw in black, and we had those thin filaments that were kind of interspersed with the thick filaments. If you remember, we had the thin filaments interspersed. So this is just detail of one of these thin filaments, right? So all the heads were on both ends of the sarcomere, and the thin filaments were overlapping with the thick filaments at the level of the heads. And you had a bare zone in the middle where you didn't have the thin filaments, all right? So the bare zone would be over here, where the thin filaments are not coming. Okay, So what happens next, once this cross bridge is formed, the heads of the mycins are going to flex. That's when they flex. After the binding into the G-actin, they flex. So think of my two arms like this. The forearms are extended. And once they bind onto the G-actin, there's going to be flexing. So following the cross bridging, you have flexion of these heads. And this happens at both ends. So I'm going to draw in black the movement of flexion of these heads. So once the heads, after they extended and bound, then they flex. So let me draw this flexed head. I'm exaggerating a little bit here, but that's okay. So these heads, once they flex, think of it as a ratcheting movement, and this is happening on both ends, right? We have the heads flexing at both ends, and what these heads are doing is that they are pulling on the actin filaments. They're being pulled inwards. So within each sarcomere, you have these filaments that are being pulled inwards, over and over. So once there is this flexion and the filament got pulled in, a little bit, the heads then dissociate from the G-actins, and ATPase kicks back in and cocks the heads back out. So the ATP is used for the cocking motion, so we can form a new cross bridge. So, so think of it as, so we have the extended positions, clipping on the G-actin, the flexing of the heads, then you have on-clipping of the heads, and then you have the ATPase that will provide the energy to re-extend those heads and put them in the cock position. So they can then, I want to show you why. So once these heads come out, you see how the filament is lined up one over. So the heads are basically kind of like clipping into the next one behind. And that's how we ratchet this thin filament inwards and inwards and inwards. All right. So, okay, this happens, and then you shrink the muscle cell kind of like inwards, all the sarcomeres kind of shrink inwards. And because you have all of these linking proteins, the whole cell shrinks, and all the cells are attached to each other. They all kind of bring this whole muscle inwards and shorter. And that's what we call contraction. So of course we couldn't stay in this position forever. We need to have a relaxation stage. So I stopped adding numbers, but the coupling stage was number 2. And then contraction was number 3. And then the last phase is relaxation, number 4. Okay, so the enzyme that's involved in this step is acetylcholine esterase. Every molecule most molecule that ends up with A's are enzymes. So that's an enzyme that is going to break down acetylcholine, which means that acetylcholine can be can stop. So if there is no more acetylcholine in the synaptic cleft because it's broken down, acetylcholine esterase is going to destroy the acetylcholine in the synaptic cleft. So it stops binding on these ligand gated channels, right? So there's no more acetylcholine binding on them. So if there is no more acetylcholine, we're going to have the ligand gates closing, they close. So no more action potential, right? We do not have the depolarization of the membrane anymore. If we close all of these channels, there is no more ions coming in, depolarizing, there is no more excitation. So it's time to pump the calcium back into the SR. So here, because we need to pump the calcium against their gradient, we force them into the SR, right? So this was the SR, and there's the pump, and the pump uses ATP to push these calcium ions back into the SR. So we need ATP to have relaxation. We needed ATP for the caulking of the myosin heads, and we need ATP for relaxation because we need to pump the calciums back into the cell. Acetylcholine will also be reuptaken and recycled by the neuron that delivered it. The acetylcholine monomers will be recycled and basically rebuilt. So a little clinical correlation with rigor mortis. This is a field that I really fascinates me in forensic science and the field of autopsies. That would have been definitely a career path I would have chosen as well. There's just so many things in life to do that are just exciting and interesting. So here, rigor mortis can be explained, can be understood if you understand the basics of muscle contraction. And that's why it's so nice to know normal anatomy, normal physiology. So when something doesn't work the way it's supposed to, it's logic that you result the result is seeing the disorder or condition. Here we're looking at rigor mortis. So this is not a disorder, this is just a result of the person having passed away, where you end up with stiffening of the body, and that's because the muscles are contracted around 3 to 4 hours after death. How can the muscles contract 3 to 4 hours after death? The person is dead. And then it peaks at 12 hours. You have like all these muscles contracting and no relaxation. And finally, relaxation takes place over the next 48 to 60 hours. How can that possibly be? The person is not alive. That's because the sarcoplasmic reticulum deteriorates, and it's one of the first things that deteriorates post-mortem. So you can look kind of the patient is the deceased is on the ground and passed, but and everything else looks normal still. Like there's no decomposition, right? It's just like right away, the body is normal. The first things that start decomposing, basically disintegrating, is the sarcoplasmic reticulum. So immediately after death, sarcoplasmic reticulum just falls apart. And what does the sarcoplasmic reticulum contain? Calcium. Calcium is released from the SR. And this calcium is going to bind to all the troponin because it's there. It didn't disappear yet. Troponin, tropomyosin complexes are there. So they're like, okay, let's do one final round of contraction. And that's what happens. Post-mortem, you have all this calcium release. You have one final contraction of the muscles. they form the cross bridge, they create the contraction, and they don't release because you need ATP to have the heads of the myosin cock back out to bind again and continue the ratcheting. Here, it's just one contraction and that's it. That's it. And it's stuck there because there's no ATP. ATP comes from cellular respiration. There is no more cellular respiration. The person is dead. So there is no ATP available. No ATP available to do the relaxation. Okay, so how come? So peak at 12 hours, that means by 12 hours, all of the muscles are contracted, all of them. And usually it happens in the position the person died. So they're usually kind of stuck in that position when they're found, a few hours post-mortem. How come this rigor mortis diminishes over the next 2, 3, 4 days? We just said there's no ATP. How can there be relaxation? So there's actually no relaxation per se. There's no the standard process of relaxation. This is not what's happening. When rigor mortis starts diminishing after 48 hours, it's because the filaments are disintegrating and they're basically coming apart from each other because they're disintegrating. It's basically decomposition that's just going further now and into the filaments themselves, not just the SR, but now the filaments are also, disintegrating, and so if they disintegrate, the cross bridges are gone. There's no more cross bridging, and it's like there's just no tightening anymore. So this contraction at the neuromuscular junction can be affected by several things, and these are some examples of toxins that are used to produce certain effects. And so I thought it would be very interesting to talk about these because you might or might not see them in a pathology course. So I thought it would be interesting to talk about them here. Spastic paralysis. This is something that can be caused by pesticides, choline esterase inhibitors. So this is the inhibitor of the enzyme that breaks down acetylcholine. Oh my goodness, that means acetylcholine is going to stay in the synaptic cleft and keep binding on the ligand-gated channel. So you end up with a continual contraction of the muscle, and you're like in tetanus, basically. So the word tetanus, I used it just as an analogy to the actual condition of tetanus. But all the muscles are contracted, so you're in paralysis because you can't move because your muscles are contracted. And spastic is referring to this contraction, and you're paralyzed because that's all you can do. You're just locked into this contraction, and you can't relax because the acetylcholine is constantly there. There is no relaxation phase. It is not recycled. No relaxation phase. Flaccid paralysis is the same outcome. It's still paralysis. You cannot move your muscles, but not because they're contracted. It's because they can't get contracted. So now it's flaccid, means kind of soft, like there's no activity going on. So curare is a poison that used to be used by Aborigine populations in the Amazon, I think. It's a poison they would put at the end of their arrow tips. So when they want to, kill their opponents, their I forgot what it's called, but the person that is being killed gets the curare into their body, and the curare has the property of looking like acetylcholine. So it binds on the receptor of acetylcholine, but it actually doesn't open the channel for the sodium ions to come in, depolarize the membrane, and create contraction. So the receptors are all bound, but they're all locked closed because the channels are not opening. So that's what curare does. It competes with acetylcholine by basically blocking the receptor portion and preventing the channel from opening. So the muscles they can't be stimulated. They're very limp. And of course, their muscles of respiration are very important to keep you alive. And this can lead to respiratory arrest. So it's a horrible death. And tetanus is this kind of third type of toxin that I wanted to share with you. It's another kind of spastic paralysis. So I don't know if I should have done them one after the other here. This one is caused by a toxin that is released by a bacterium called Clostridium tetani. These bacteria, once they kind of infect you, they release this toxin, and this toxin overstimulates the muscles and they overcontract, and you're like, you're just like locked up into this spasm. The muscle is constantly contracting, and it can literally like break the bones of your back, because it's there's some images of individuals with tetanus, and their hands are locked up and their back muscles are all locked up, and it's really terrible. Another clinical correlation, because you probably will see that one for sure, myasthenia gravis even included there's new research about a potential treatment for myasthenia gravis. I shared with the documents I shared with you. So this is an autoimmune disease where antibodies of your own body are attacking the neuromuscular junction. So the neuromuscular junction sees the acetylcholine receptors completely clustered together, instead of being, I think I drew them in green, the ligand-gated channels, remember? I think they were in green and the voltage-gated channels were in blue. So these acetylcholine receptors are literally kind of clustering together. They're kind of clustering together on the surface of the membrane, instead of being all kind of spread out and all being able to receive an acetylcholine. These antibodies are basically just kind of like coating them and clustering them, and they become not available for binding of the acetylcholine. It's like a big jumble of molecules on the surface of the cell because of the antibodies attacking the surface. So the acetylcholine can't bind. So it's the surface becomes less and less sensitive to acetylcholine. So the muscles are less and less excitable. So they're just more limp. And the first muscles that get affected by this condition are levator palpebrae. So you have a drooping of the eyelids, and the muscles that hold the eyeball are also kind of relaxing. They're just not, because they're not contracting, they just kind of like go limp, and the eyeball shifts. You end up with double vision, the eyelid droops, and then muscles involved in swallowing get affected. So now you have difficulty swallowing. It's got to be a hard feeling. And then your limbs become weak, and then eventually it's respiratory failure because of the muscles of respiration. So if it's caught early, if you have a physician if there's a physician that hopefully you in the future can see these signs of myasthenia gravis and catch it early, there are certain treatments that are available now. with cholinesterase inhibitors, basically to increase the availability of acetylcholine in the synaptic cleft there at the neuromuscular junction, or thymus removal to kind of diminish the number of antibodies produced, or just literally giving immunosuppressive agents to kind of like, calm down the immune system, because this is an autoimmune, so it's your own immune system that's kind of creating this problem that doesn't exist. It's really, really kind of frustrating. All autoimmune diseases are kind of frustrating diseases because it's your own body reacting to yourself. That's it. So this was the last slide. I wanted to talk about a few conditions that relate exactly to what we talked about in this chapter. So thank you for paying attention, and goodbye.